my name again is Tao. So right now I'm in a research-based master's program right now in the Department of Physiology, and my supervisor is also Dr. Mansoor Hussain. So a big thank you to Dr. Ju for inviting us here to speak today. I'll be telling you a little bit about myself and how I got started in research, so starting off with that, before talking about, like the title says, vascular smooth muscle progenitor cells in cardiovascular disease. Yeah, so right off the bat, just no conflicts of interest to disclose. So for me, I am originally from Ottawa. And then I lived there all my life, and I came here to do my undergrad at UOT. And so, like most of you, I also did HMB, so I did a health and disease specialist with a physiology minor. And also, like most of you, I took Biochem 210, Bio 230, Bio 220, like PSL 300, 301 again, in my second year. And I don't know if some of you relate to this, but I didn't do too well during that second year. I had a really rough year. So by the end of the year, I was still kind of like, what am I doing with my life? What am I doing with my degree? And a lot of my friends walked out of those courses being like, oh my God, I love immunology. I love like the kidney, things like that. But for me, like that didn't really click for me. So I'm studying cardiovascular disease right now. But looking back, when I did take like PSL 301, we would always see things like cardiovascular disease is the leading cause of death worldwide. Or they would throw this picture right here, which is the cardiovascular system. But the thing is, when I saw this, I would always just kind of be like, okay, these are like numbers and pictures on a page or on my PowerPoint slides. It doesn't really mean anything. Still trying to figure out what I'm trying to do. So right after that, after my second year, I went home to Ottawa for the summer, and I was trying to find something to do that was life sci related. I was like emailing profs, emailing labs, trying to look for jobs, things like that. Ultimately, I got a really good opportunity at the University of Ottawa Heart Institute with Dr. Franz Lehnen. And so for me, that was pretty life-changing, just because I got to see a lot of really interesting guest lectures, got to see a lot of the different research that was going on, and learned a lot about the new ways that people are trying to diagnose heart disease, treat it, or prevent it. And so it felt like I got to put a name to the face almost, and I really got to see what I was learning in class hands-on. So for the work that I did in Ottawa, I won't get too in-depth in this, but I got the opportunity to work on a project looking at how exercise increases this thing called FNDC5 irism. And that in turn increases different factors that are beneficial for the heart and brain. So I've included these two papers here in case you're ever interested. And I think Dr. Ju likes to talk about this during some of his courses, so just keep an eye out for that. And for me, I got a really cool opportunity to actually write up my work as a paper that is being reviewed by a journal. It actually just got turned down recently, but that's okay because we're just gonna fix it up and then resubmit it, and then hopefully it'll be okay. Oh, and then I just have this cool video here. I don't know if it'll play. Yeah, so that was just the rats that I was working with. They were running on a treadmill. You just put some peanut butter on their food, and you stick it at the top, and then they just run towards it. And they just keep running, and then they end up really fit. I think I actually named these guys Usain and Bolt. So that was a kind of fun time. But yeah, so for me, afterwards, I kind of realized how interested I was in cardiovascular research. And so I enrolled in HMB422, so shameless plug right there. It's a really good opportunity. But I managed to find a position at SickKids looking at heart regeneration with Dr. Paul Delgado-Olguin. And so kind of the background for that is just that when mammals are newborns, we're able to recover from heart injury if it occurs really early on. So like in the first week after birth, mice can regenerate their hearts. And then same with pigs. And there's actually been some reports that it's happened in humans as well. So that's pretty interesting. And again, I'm not going to go into too much detail in that for this, but we were thinking that it was due to differences in the developing heart before and after someone was born. And so my project was looking at whether certain stop signals that are involved in development might be able to kind of be harnessed and manipulated to control heart development and try and return the heart back to its regenerative state. Ultimately, not all findings are groundbreaking, and we actually found that this gene that we were studying was not involved in this whole process, but still it was a really great opportunity to do some cool research, and we actually managed to publish it, which was kind of nice. So these two experiences kind of opened my eyes to what research is like. So now when I look at this picture, what I think is that the cardiovascular system is really widespread, and it connects basically every part of the body. And because of that, whenever you have a problem in your cardiovascular system, it can lead to problems everywhere else. But that's also really exciting because it means there's a lot of other new perspectives that you can gain from studying other systems and new approaches that you can do it. So new ways to think about it, new ways to study it, and new ways to treat it. Also recognizing that cardiovascular disease encompasses a lot of different things. So on the cardio side of things, that might include cardiac arrest or myocardial infarctions, which is an actual blockage of the heart, and ischemia. And then on the vascular side of things, you can have cerebral vascular disorders like stroke. You can have microvascular dysfunction, like during diabetes, which ultimately results in the loss of blood flow to things like your eyes or your feet. There are also aneurysms, which are a weakening and bulging of your blood vessels, which can be potentially fatal when they rupture. There's also atherosclerosis, which is the buildup of fatty plaques in the blood vessels. And then this can ultimately lead to heart attacks as well as stroke. And when you're actually trying to treat these though, you can actually cause more problems. So sometimes by putting in a stent to prop open the blood vessel when it's narrowing or getting blocked, you can actually damage the inside and then cause a sort of proliferative response inside that'll narrow the artery again. So a narrowing of something like an artery is called stenosis, and then after you open it up and then if it recloses again, that's called restenosis. So for the most part, my graduate work is focusing on more the vascular side of things, so more so the stuff in the red box. And specifically more so diseases in terms of their more so vascular remodeling diseases, so anything that's affecting their structure or their function. So just a quick overview of what exactly vascular actually means there. So some of you might have been exposed to this in HMB422 or in anatomy, but basically, if you recall, the artery wall consists of 3 different layers. So there is the innermost intima, which has a lot of different endothelial cells. There's the middle layer, which is the media, and this has a lot of VSMCs, or vascular smooth muscle cells, which is something that our lab has been focusing on for the past 2 decades. And there's also the outermost layer, which is the adventitia, and there's typically a lot of fibroblasts and extracellular matrix in there. There's also immune cells floating around here and there inside, but it's also important to note that there are actually stem cells, which I'll be referring to as vessel-resident progenitor cells, inside the blood vessel wall, and that's actually responsible for generating these different types of cells inside. So focusing on these VSMC, we know that VSMCs are critically involved in vascular health and disease. So while the rest of the layers are a little bit more flimsy, that thick media layer is really important because it provides not only structure to the blood vessel, but because it's muscle, it also contracts and relaxes to facilitate changes in vascular tone, so like vasodilating, vasoconstricting in response to different things. And ultimately, because of how important VSMCs are, if there's a problem with VSMCs, then you often have a problem then you often have vascular remodeling cardiovascular disease. So for example here, on the left, that is an example of a healthy mouse aorta. So kind of at the top, that's where the heart would be. It would branch off. The little things at the bottom are the arteries that go into your legs, and then the little two parts above, that goes into your kidneys. But ultimately, that part where the arrow is pointing, so in a healthy artery, you just see a big long tube. But then when you have an aneurysm, it bulges out. And then if you take a cross-section right where that arrow is, then what it will look like is, on the left there, you can see in the red, it's calponin, which is a VSMC protein. And so whatever's in the red, those are your VSMCs. So you can see that it's pretty, it takes up most of that vessel wall, and it's pretty well distributed and organized. When you have an aneurysm on the right there, you see there's a lot less red, and you see that it's a lot more disorganized, and that ultimately will lead to a lot of different problems. So it's pretty clear that these VSMCs are pretty important. But one thing to also think about is, what about their progenitors? So before you can have these different mature endothelial cells or the smooth muscle cells, which are contributing both to health as well as disease, you need the cells to first be produced by these progenitors. And so looking at progenitor cells, our lab wanted a way to study these progenitor cells and then be able to get them from humans. So we did a study using progenitor cells from human skin. These are known as skin-derived precursor cells. And what you can do is you basically just take a skin biopsy, you digest it into a single cell mix, and then you throw that in a dish, and then they'll form little clusters, and then from those little clusters you can get skin-derived precursor cells. So when we took skin-derived precursors from patients with type 2 diabetes, we actually found that patients with type 2 diabetes had fewer progenitor cells from their skin, just to begin with. So there's something wrong with the progenitors and the lower number. When we give them different factors that cause them to differentiate into VSMCs, we noticed that these VSMCs also didn't work properly. So one way to kind of test whether they work or not is using a wound healing assay. So this is a way to kind of just streak a line down the middle using a pipette tip or something, and then this creates a sort of quote-unquote wound. And ultimately what it should do is that after you create that wound within 24 hours, the cells should kind of repopulate it and grow back into that wound to heal it. But when you take VSMCs from progenitors that were derived from patients with type 2 diabetes, it didn't do this properly. So not only were the progenitors themselves affected, but it also carries on to the different cells that they actually ended up producing. The downside to using these models, so getting these cells from humans, which is really nice, is that these aren't the progenitor cells which are actually found in the blood vessel wall. So for my graduate research, I'm mostly looking at these different progenitors which are marked by a protein known as stem cell antigen 1 or SCA1. So these SCA1 progenitors have this protein that no one really knows exactly what it does. And because no one knows what it does, it's been really hard for them to kind of find the same thing in humans, which is one major caveat. But the cell surface protein it's on a lot of different types of cells, a lot of different types of progenitor cells. And so even though we don't know what it does, people are still using it because it's a really good marker of different progenitor cells. And there's a lot of different types of cells that express SCA1, but we can isolate the ones that specifically produce VSMCs using a number of different markers. So immune cells also express SCA1, and then by kind of excluding anything that expresses an immune marker, then we can figure out exactly what is immune, what's not immune, what's progenitor derived, what's not progenitor derived. And so lastly, these SCA1 progenitors are really important for health and disease. So the first study which ever identified these cells found that in the aorta, they found a lot of these different SCA1 progenitors in the fatty plaques that form during atherosclerosis. So these VSMC progenitors have been kind of investigated by a lot of different groups, including ours. And so since then, a lot of people have looked at how they might contribute to different disease states. Something that our lab did was actually look at what's happening during development. So whether these SCA1 progenitors are actually involved in the development of the different blood vessels. So for us, we looked at when and where during development, and whether these came from development at all. And into adulthood, one of the things that we looked at was where they came from. And so one of the cool things that we found was that starting during development, in a germ layer region called the Semitic mesoderm, that's kind of where they start to appear. And then they're in really low numbers, even after birth, but when they hit about 4 to 6 weeks of age, you have this huge increase in the number of SCA1 cells as well as SCA1 derived cells, particularly those from the Semitic mesoderm. And so it's pretty clear for us that even in a healthy blood vessel, they're contributing a lot to that blood vessel. So the majority of my work during grad school is centering around blood vessel injury. So for people, when they have a blockage in an artery or build up a plaque, you can go in with this wireframe called a stent. Some people also go in with like this deflated balloon that you can slide into the artery, and then you expand the balloon and it basically just blasts it open. That can have a lot of different complications associated with it, so I think most people have been going with the stenting lately. But ultimately, the fact that you're going into this artery with a foreign object is going to cause some sort of injury. And what that injury leads to is inflammation, so the recruitment of different immune cells, and loss of cells because you're physically scraping off cells when you go in there. Ultimately, what that causes is a proliferation of the cells that are inside the vessel wall, and what will happen sometimes is those little holes on the side, that's where the stent was. And then what will happen is the cells are just like, we don't care about the stent, they'll grow back into it, and it forms what's called the neointima, so new tissue on the inside of the intimal layer. And this is a process known as neointimal hyperplasia, and ultimately leads to that restenosis of the artery. In our lab, we use a surgical model known as the wire denudation injury to kind of model this process. And so we look at the mouse common carotid artery. So what we can do is we can open up the neck, expose the carotid artery, and then we go into it and we use a medical guide wire to kind of scratch the inside of it to cause that sort of mechanical injury in the artery. And then ultimately what we end up having is this. So on the left there is an uninjured artery, and then on the right, you end up with that neointimal hyperplasia, which grows in there. Though I think that this picture is a little bit more of an extreme case, but you end up with a similar effect. So some of what our lab has been doing is looking at these SCA1 progenitor cells in different vessel injury models. And so some of the work that I've been doing has been looking at cross-sections of these, basically doing the injury, taking cross-section, throwing some antibodies on there that look at specific proteins, and then throwing some more antibodies on there that are fluorescent, so they'll bind to the antibodies from before, and then you can label different proteins here and there within that artery. And so when you look at the green there, that green is the SCA1 positive cells in that artery. So on the top, you see it at 8 days after injury. So there's some green cells, they're starting to move around, they're starting to proliferate. And then at 14 days, you have a massive amount of green there. And so one of the things that I've been doing is trying to define exactly what types of SCA1 cells are contributing and where. And so, for example, in the red, I stained for CD45, which is a marker of immune cells. So by looking at these, I can kind of look at what is immune, what is SCA1, what is immune and SCA1 positive, what is immune and SCA1 negative, what is SCA1 positive and non-immune. And then that's on the graph on the side there. So the shaded region is anything that is non-immune but SCA1 positive. So ultimately, you see that there's a lot of shaded region there. So most of these SCA1 cells that are proliferating are progenitor derived, and it's inside that vessel wall. So the take-home message here is that these SCA1 progenitors are really important and that they're facilitating a lot of what's going on there. And that's not to say that the immune cells aren't doing anything, but what it looks like to us is that these progenitors are actually doing most of it. And so adding on to that, our lab has previously looked at what happens when the SCA1 progenitors don't work. So Luke introduced the CMYB hypomorph mutant, and so those mutants reduce expression of the transcription factor CMYB. So that also regulates VSMC activity. And so interestingly, when what was done in the past is that they went in and then they did that wire injury on these CMYB hypos. And then they found that there was less new intima. And that was really interesting. So then they started looking at what was going on, why was that the case? And then they stumbled upon something really interesting, because when they looked at the progenitor cells, they found that in terms of numbers, these progenitor cells were not effective. But when they tried to differentiate them and make them proliferate, it didn't work. So this was really cool because it showed that SCA1 progenitors, when they don't work, there's no injury response after. And so it was kind of interesting because that means like maybe if you target these cells and you stop them from proliferating, you actually don't have this restenotic remodeling effect. So a lot of the times with research, when you go and try to answer one question or answer a few questions, a lot more pop up, but that always leads to more research. So a bunch of the questions that we've been thinking about recently have been things like, if SCA1 progenitors are also dysfunctional in disease, because no one's ever really looked at that. So one of the advantages of looking at these SCA1 progenitors in mice is because you can generate a lot of different mouse models of different diseases and then study the cells from them. So you have this really efficient way of controlling for that environment and kind of modifying that and then studying these cells directly from the blood vessel. We also know that since these SCA1 cells are really important, and if they don't work properly, then you can control that vessel injury response. But is there a way that we can target these progenitors specifically? And a lot of different studies recently have been looking at, like Luke mentioned, without targeting a specific cell population or something like that, look at what they're doing, look at what's controlling them, what's affecting them, and then target that. So those are a couple of different approaches there. So some things that people have looked at have been like things like cholesterol or microRNAs, which kind of try and modify some of this. And then lastly, I mentioned that no one really understands what SCA1 is doing. That's something that I've been focusing on recently, and I'll just quickly show you some of the work that I've been doing for that. So right now, the best guess that people have is that SCA1 expression controls progenitor function, but this is really just based on a couple of papers here and there, a couple of studies, and a lot of them are just associations. So generally, people think that if you don't have SCA1, like if you have a transgenic mouse, mutant mouse, that is deficient in SCA1, then you end up with uncontrolled cell growth. So what 3 groups have actually shown in the past is that if you have a mouse that does not express SCA1, then they have bigger hearts. So progenitors in the heart are a pretty controversial area, so I'll just kind of leave that as food for thought. But on the other hand, if you have too much SCA1, then there's no expansion of cells. So this graph that I'm showing, basically what it's trying to show there is that the top graph is mouse bone marrow derived hematopoietic progenitors, and then the bottom is human bone marrow derived hematopoietic progenitors. And then when they overexpressed SCA1 with an adenovirus, these cells no longer exhibited progenitor-like activity. So when you grow them on a plate, typically they should kind of aggregate into little cell clusters. but in this case, they did not. So no SCA1, too much cell proliferation, no SCA1, too much cell proliferation, sorry, and then too much SCA1, no expansion. And based on that, the question I've been asking is whether SCA1 expression is required for vessel injury response. So right now, the answer seems to be yes. So I've actually gotten some of those SCA1 knockout mice myself. And because I know that immune cells express SCA1 and I can use that as a positive control, I did what's equivalent to almost like a blood test on these mice. So I took some blood, ran them through a technique called flow cytometry. And so basically anything that is in that box is SCA1 positive, that little blue box right there. So whenever cells kind of land there on the profile after it looks at the different fluorescent levels. So we go in with a fluorescent conjugated antibody that labels SCA1, and then based on that fluorescence, we can look at whether it's SCA1 positive or negative. So on the top, you can see that the WT is normal expression, it's from a wild type mouse, and then below there's negligible expression in the knockout. So these are true knockouts. And then when we did the wire injury, we stained it with Massen's trichrome, which Luke talked about, and then we looked at uninjured and injured blood vessels. And so long story short, it looks like our mutants actually have larger blood vessels at baseline compared to normal wild type controls. And then after injury, the vessels become much larger. And when we looked at the different layers, this was specifically in the adventitia. So what we're seeing here is, and what we're thinking is that there's more scar tissue and more fibrosis that's popping up after injury. So there's some sort of thing being misregulated that's causing more fibrosis. And then on that same note, when we looked at cell proliferation to kind of see, are these progenitors differentiating more, are they proliferating more? We used a marker called Ki67. So when cells undergo cell division, they express Ki67. And we found that these knockout mice after injury actually have more proliferating cells compared to normal wild type controls, which is consistent with the idea that SCA1 might be something that's keeping these progenitor cells in check. So just to kind of close off, I want to thank Dr. Ju again for having us, and huge thanks to everyone in our lab and anyone who's been helping me, as well as our funding sources, and thank you. Thank you for listening.
So Tao, could I ask, why didn't you stay with the same supervisor you were doing your original fourth year project with?
So I think, it's a bunch of different factors there. I think for me, it's always nice to kind of diversify and then get a lot of different research opportunities. So I think one of the advantages of undergrad is that you're not committing to something in terms of like, if you're doing a PhD, you're committing to a lab for like 4 or 5 years. Undergrad is a nice time to try different things. That was one way I thought about it. Also, when I actually applied to a lot of different grad departments, and not all of them took me. So the lab I was in for my 499 project was in molecular genetics, and unfortunately I didn't have much of a genetics background, and they took that into consideration and I wasn't accepted into that program. But I was really fortunate that when I was looking around for labs, I found a lot of different opportunities, and I got to kind of pick the ones that I was interested in and then look for those. Yeah.
What do you plan on doing after you've completed your
So I'm planning on doing a PhD, and then I'm thinking either academia or maybe, like Luke mentioned, also a clinician scientist position. That's like really far off. So I think I'm just, for me, I like to kind of take things one step at a time. So I'm just thinking PhD and then exploring other options there too. That being said, I'm also pretty open-minded because there's a lot of different career opportunities I didn't know about until I got into grad school, and then I started talking to people. I got to see like, what are people doing? What are different companies offering? What are the different options out there? You get exposed to a lot more of that later on. So I think I'll just like kind of keep an open mind too.
Sorry, at the beginning you mentioned what kind of master's program you were in, but is it course based?
It's a research based.
Oh. What's the big difference between course based and research based master's programs?
I would say, so research based, you have to write a thesis at the end, and it's more so that you're in a lab, generally from like 9 to 5, maybe longer, generally longer, and then you're basically working towards ideally publishing, working on that thesis, writing up, and then being able to show your results and show everything you've done during that. There's less emphasis on courses, so it'll vary by different departments. So for physiology, we're required to take 2 half courses for the master's and then 3 half courses for the PhD. So that's because they also want you to focus on your research, but also kind of keep up with what's going on in the field through those courses and train your critical thinking skills through like discussion and stuff. So those ones are generally more seminar based. And then course-based masters, I'm not sure if I can speak too much on those because I'm not too as familiar, but generally they're kind of like what you're going through right now with like the different courses, and yeah, I think like I would talk to the different grad departments and then find out more about it. And I think there's also like a graduate research fair as well, so really great question to ask that you could direct to them.